Okay, well it's a great pleasure to welcome Cheryl again for a second talk, who nobly stepped in at quite short notice for us, for which we're very grateful. As you've heard, but I'll say again for the camera, Cheryl has been at Exeter, where she did a PhD in cell and molecular biology. Then a few years ago she did an MSc in theology, and for the last 3 years she's worked in the theology department on a range of issues actually, involving ethics, the environment, and some of the things she's going to talk about today. So thank you very much indeed for coming, and we look forward to your talk.
Thank you. Before I get engrossed in the talk and forget, I would like to suggest one book which you might find helpful. Covers several of the topics we've looked at today: genetic engineering, in vitro fertilization, stem cell research, and it's a book by a colleague, or two colleagues of mine down in Exeter, John Bryant and John Searle, called Life in Our Hands. It's been out for a few years now, but it's still a very good, very useful resource, especially if you're new to this particular field of research. So that's on the bookstall at the moment. Right, well, what we're going to do in our last session today, thinking about ethical challenges in contemporary science, is focus on stem cell controversy. And obviously there's going to be a deal of overlap in topics between this session and both the previous two, in that the technology, the development of the technology to combine sperm and egg outside the human body and to grow the early human embryos in petri dishes has enabled the development of other technologies, including the growing of stem cells and including the pre-implantation genetic diagnosis that we discussed briefly in the last session. So there's a degree of interaction here. I'm going to take a slightly different tack in that I'm going to talk about what's involved in terms of the science, but then also focus on different approaches to do with religious reflection on the topic and also the development of public policy. So what we're going to do is look at the what, where, why, how stem cells first. I appreciate that many of you will know this already, but please bear with me because some people don't, or said they would appreciate going over a little bit of the theory again. So please bear with me if some of this is very familiar to you. Then we'll look at the controversy surrounding stem cell research and some of the reasons for that. And then we'll look at some of the strategies that have been used in the public discourse on this subject. And as you're aware, probably, certainly in this country, it's been a very hot topic with the passing of amendments to the Human Fertilization and Embryology Bill last year. And I suspect in many countries there have been legal situations arising to do with stem cell research, and there's certainly provision in the laws of the different European countries and in the States on what may and may not be done with human stem cells. I should do first. Actually, let's just go back a minute. What I want you to do is forget about the pictures for the moment. I want you to sort of have a thought experiment, as it were. I want you to imagine that the human egg is like a very big, very well-equipped, very well-stocked kitchen. There's every implement you could dream of for cooking. There's every ingredient you could require to make a human being. And there's half the recipes in the human egg before fertilization. And then the sperm comes along and provides, as I'm sure you're all aware by now, the other half of the recipes, half the genetic material from the mother and half from the father. So you have, if you kind of picture in your mind's eye this kitchen with all these recipes spread out. And that's everything that you need. And as we've heard earlier today, as that cell divides and develops into a human being, as the cells specialise, we still have that same set of DNA in each of the cells of our body, except the very few that specialise to actually not have any nuclear material in them. But most of the cells in our body have that same set of DNA. What makes them different, what makes a bone cell different from a blood cell or a skin cell, is not the DNA that's in it, but the DNA that is expressed within it. Some of those recipes are being used still, and some of them have been put away. So imagine your recipe book has been shut, if you like. They're still there, they're still within the cell, but they're no longer being used because the cell is specialised and it only needs a certain number of those genes. So we've got this fertilised egg with the DNA, all the recipes there, the potential to grow any and all cells in the human body. And that's what we call a totipotent cell. It's able to grow into a full human embryo and with the support tissues, the placenta and support tissues that enable it to grow within the mother's uterus. Now, when you imagine, and we'll just flick onto some pictures first. So at the top here, we've got a human egg surrounded by sperm. This is a very famous set of pictures that was in the journal Biochemistry, I think, a few years back. Surrounded by sperm, and one of the sperm gets in and fertilises the egg. It's a very large cell, as cells go, egg cells always are, because they've got all the goodies to start development off without any other nutrients coming in, because for the first few days of development, the developing human being is on its own, as it were. It's in the mother's body, but it's not implanted in the warm wall of the uterus. So it has everything it needs in the egg to start with. And then sometime later that cell divides to give 2, you don't need me to go 2, 4, 8, etc. And you get a ball of cells, progressively smaller but more in number until you can't actually see the individual cells very clearly. Now at this stage, they're still capable, they've still got all those recipe books open. They're still capable of forming all the tissues in the body. They're so flexible in that respect, in fact, press the wrong button, that if you take one, as we saw earlier in John's talk, if you take one of those cells out to test it, for instance, if you want to test to see if the embryo is carrying a disease and you want to make sure that the embryos you implant back in the mother's womb are not carrying the disease, you can take 1 or 2 of those cells away, and the ball of cells that you have left will still grow into a perfectly normal and healthy embryo. You don't need all the cells to be there, because they've still got that, they're still totipotent, they've still got that ability to grow into the whole baby. And it's also at this stage that there's so much flexibility in the early embryo that the ball of cells can even be split in half, and you can get identical twins. Similarly, there's so much flexibility that occasionally, we don't know how often it occurs, but occasionally you'll know people who have non-identical twins. So there's not been an embryo splitting there. Non-identical twins are where the mother produces 2 eggs in the same monthly cycle, and if they both happen to get fertilised by sperm in the same cycle, and they both happen to get embedded in the wall of the uterus, then the woman will have 2 non-identical twins. But occasionally we've become aware that 2 non-genetically identical progeny, like that, 2 non-identical twins, sometimes at a very early stage in development, those 2 balls of cells, if they come close together, can actually fuse. And the resulting ball will then form one baby. So it's the reverse of natural twinning. If you split this in half and you get 2 genetically identical twins, you imagine the reverse happening. 2 non-identical twins can actually fuse to give 1 human embryo, and then you get an adult who actually has cells with 2 different genetic identities in their body. We don't know how often this occurs, but we've discovered with things like paternity testing, it's occasionally thrown up a case where the mother is apparently not the mother, never mind who the father is. The mother's not been right, and when they've looked, they've taken a sample, maybe a swab from her, mouth to get her genetic profile. They've discovered that it's not the same as the reproductive tissue, and that the egg she's produced has a different genetic mother, if you like, than the cells, the tissue that they took to test her. She is a chimera of 2 genetic identities. We have no means of knowing how many chimeras there are, because most of us aren't going around getting DNA tested with different bits of our body. But it's just to illustrate the flexibility that there is at this stage of human development. So we have, sorry, keep pressing the button. We have at this stage a huge amount of flexibility. We can take 1 or 2 cells out and no harm is done. Natural twinning can occur. If we're handling in the laboratory, if we're handling animal embryos, we can create artificial twins. If we're wanting to grow animals and clone them, just create artificial genetic twins. At this stage, the cells will still grow into a perfectly normal, if it's an animal or human. I wish somebody would invent a word for a born person. You know, there's words for sort of this embryo and there's fetus and there's neonatal, but sometimes I wish there was a word for a born person. The embryo can grow into a normal adult eventually. So there is this flexibility. At some point, and this is just a list of stages in development, we've got, this really just illustrates some of the early stages. You've got sperm penetrates the egg. It takes a while for the genetic material to come together. These are all approximations of how long it takes. Maybe 36 hours before we get the first cell division. Then you've got that ball of cells. And then at some point, the ball of cells actually forms an inner cell mass surrounded by another set of cells. And if I just show you again, this is this stage here, stage 5. And this is the stage, these cells here are going to form the embryo, and these will form the support tissue. But it's these cells which are used as a source for human embryonic stem cells. And I'll explain what I mean by that in a moment. So this is the stage with which we're concerned in this particular talk. But I just wanted to do this bit first to give you an idea of where we're at and the fact that the fact of this kind of plasticity, if you like, in the human embryo at that stage. Now, when the cells in the developing human start to specialize, the biological term is differentiate, they start to specialize and form cells forming particular tissues within, firstly the embryo and then the developing baby and the adult human. And at this stage, these cells, if you take these are not as flexible as these, but they can still give you all the cells in an ordinary body. They can't give you a whole embryo, but these cells can divide to give possibly the outside cells in your body, the ectodermal tissues, the cells lining your gut, the endoderm, and the stuff in the middle, the mesoderm, the bone and blood and other connective tissues. So whereas we call, sorry, I'm never very good on technology, whereas we call these cells totipotent because they can give any cells forming the embryo and support tissues, we call these pluripotent. So totipotent, all potent, pluripotent, a plurality of potencies, but not, you can't actually grow a human embryo from one of these, but you can give any of the human different cell types. So pluripotent. Now moving on to our experiment, it's as if these cells have some of the recipe books have been closed, but a lot of them are still open. As the embryo develops and you start to see something that looks a bit like a human, and then you get a baby and then you get an adult, as the cells in my body, your body, specialize, gradually they close up more and more of those recipe books. The recipes are still there, but they close up the books. But there's still tissues in your body, like bone marrow, for instance, which can provide a range of different types of blood cells. And so they are multipotent. They can't give as many different types as the pluripotent, but they can still give a few. So this is the stage at which you're taking cells out for pre-implantation genetic diagnosis. This is the stage of embryo at which you'd be handling in the laboratory before implanting it back into the mother. This is the stage at which you're going to try to take stem cells from the inner cell mass. So the the cells you get in an adult body at the most are multipotent. There are some cells in the adult body which are multipotent. You can also find these in late fetal tissue, and you can get stem cells in umbilical cord blood, and I heard recently they're investigating using placenta as a source of stem cells. Nowadays, they have started in some hospitals offering a service to new parents that you can keep the umbilical cord blood from your child when it's born and store it frozen so that it is a source of potential stem cells for use if that child has a need for them medically at some point later in life. So there are some cells that are useful in the cord blood, and there are a few tissues in the adult cell which still retain a certain amount of flexibility. Got a few recipe books open, can give you a range of different cell types. Then reiterating, the pluripotent cells, that's the ones in here, can make lots of cell types, and that's the blastocyst is just the technical name for that ball inside another ball stage. Only the very, very early embryo is totipotent and can give all cell types, including the support tissues. So this is what we call stem cells, are cells which have the ability to give you a range of different cell types. Some can give you a small range, they're multipotent. Some can give you a wider range, they're pluripotent. The flexibility there is the key to why stem cells are valuable. Why are they valuable? Because they have more of those recipes open and accessible and can be directed to specialise into a wider range of different cells. Why would we want this ability? Well, quite simply, I say simply, it's a hugely complicated scientific endeavour. The idea is that if we have unspecialised body cells, we can grow specialised body parts when we need them for medical purposes. So if, for instance, somebody loses the use of an organ and they need replacement, and we talked a little bit this morning about kidney transplants and the need for donors and getting tissue matches and how difficult it is. There's never enough donors to give enough spare organs. If we could grow that patient a new organ to replace the damaged one, that would be perfect. We wouldn't have to wait for somebody to die who had a suitable match. We could try and grow them. Or another possibility would be if somebody had some damage to their body and they suffer nerve damage in their spine, maybe we could grow new nerve cells to re-establish links between their central nervous system and the bits of their body which are no longer connected due to spinal damage. Maybe some have the bioutopia imagery again. Maybe some have suggested we could effectively live forever, because whenever a bit of your body got tired and worn out or damaged or diseased, we'd just grow you a new bit. That would be the perfect solution. We're talking about the secular eschatologies earlier today. That could be another one. We can just grow you new bits of body as you wanted. So now the idea would be, one particular model for using them would be to have a bank of lots of different cells with lots of different tissue identities, so that if, for instance, you needed a new kidney or you needed a new bit of heart, we could find a good match to you in a tissue bank, and then we could grow you, we could learn how to manipulate the genetic material in that cell to grow the specific tissue that you need. We could direct the cell down a particular path of specialization, of differentiation, to replace the tissue that you want. You would still, however, as you often do with transplants nowadays from another person, from a donor, you would still probably need some drugs to make the tissue appropriate for your body. You'd have to suppress your immune system so that you didn't reject the transplant, because it wouldn't be a perfect one. Now the advantage, this is stem cells generally we've been talking about, but the embryonic stem cells are the ones where there's a real controversy, because Adult stem cells, well, you can get adult stem cells from adults, you can ask them, you can get ethical permission, but to get the embryonic stem cells from that very early stage, you have to destroy the embryo. Now, obviously, this provides an ethical dilemma for many people, not just those of different faith communities. The idea that you destroy one human life to help benefit another one, when that human life has no means of agreeing to it, is generally regarded as a deeply unethical practice. But people have wanted to use embryonic stem cells because, for one thing, they are more flexible. Because they're at that earlier stage, when there's a greater number of recipe books still open, you haven't got to worry about trying to find a cell that will differentiate into the particular type. You've got a much greater choice. You've got these very early cells. You could nudge them down a number of different developmental pathways if you learnt the chemical cues to get the DNA to behave that way, to specialise into a particular type of tissue, and you've got much more flexibility. There's one other advantage, key advantage to embryonic stem cells, in that you can clone with cells at this stage. So, for instance, if you have a patient who needs a replacement organ, you could take one of that patient's cells from their adult body, and you could take an egg cell, and you could make a human embryo to match, you could clone a human embryo to match that patient's genetic identity and grow them tissue that matched their human tissue pattern exactly. So there wouldn't be any rejection. This would involve taking one of the cells from the patient and taking a human egg cell. This in itself would need to be donated, which again has a whole range of ethical issues around it, because collecting human eggs from women's bodies is a fairly invasive and potentially dangerous process, and egg cells, human egg cells, are always in short supply. You take an egg cell from a donor. This is unfertilized, so this would be as it was from the woman's body. And for cloning, or for the proper name, somatic cell nuclear transfer, you would take the genetic material in the donor woman's egg cell out. So that's half her genetic material there, but instead of a sperm coming, we're going to get rid of that material altogether, leaving what's known as an enucleated cell. And the next stage is to put the patient's DNA into the enucleated cell. In practice, effectively, often they put the whole cell in, but just for the purposes of illustration here, we've put the whole DNA from that patient into the egg cell from a woman's body. So this egg cell now has a genetic identity that is the same as the patient who needs the tissue. And at that point, you just give it an electric shock, and this will start to divide. Normally, when a sperm enters the egg, the process is quite complex, and as the sperm enters the egg, it triggers a process which prevents any other sperm getting in, and also starts the division process eventually. But in this case, we'd have to give it an electric shock, a sort of artificial prompt. But then this would start to divide. You get 2 cells, 4 cells, 8 cells, if it was legal. If you put this into the body of a woman and treated her with the right hormones, you might, if it implanted, get a human being from that cell. And the human being would effectively be a clone. It would be a younger twin of the patient. That's not the idea here. The idea is to grow this into a ball of cells, and then, if we have the right chemical cues, if we've done the right experimentation, we should be able to direct that ball of cells to grow into a replacement organ or replacement tissue for whatever the patient needs. Can I just stop there briefly and check how is everybody okay? Okay, everybody with me still? Anybody got any questions arising on the technique so far? Okay. Yes.
Embryonic stem cells totally potent then?
No, the stem cells are pluripotent. Sorry, pardon me. The stem cells are taken when it's pluripotent. Sorry, I need a drop of water. The very early embryo is totipotent, and if you split it in half, you can get two identical twins. But once it gets to that stage where the inner cell mass is, the cells there, the cells that you take out from that inner cell mass, and I'll show you again when we get to the next photo, are pluripotent. So they'll give you all the different tissues from the embryo, but they won't individually, they wouldn't grow into a whole baby.
So can you grow stem cells from the totipotent cells?
You could, but
I believe it has been done by a few companies
yes,
in the United States.
I'm trying to
but
think.
for mouse models, not for human.
Yeah, I mean, and certainly the restrictions, there are fairly tight restrictions. I'm not entirely sure whether most of them, the models they're taking the inner cell mass cells and at the pluripotent stage and growing those. That's the, that's the model I'm familiar. I don't say I'm not familiar. Maybe they're doing some stuff in the States.
With the animal models.
With animals, but not with humans, yeah. Right, thank you. Yes, any other questions?
Do I get it right that to make often therapeutic cloning, if you have real plants fit into human?
No, sorry, I was saying that as a comparison. No, I said at this stage, if you put it, once it got to the separate cells, technically you could grow a younger twin, but that's strictly illegal, and nobody is suggesting outside the boundaries of science fiction. I think science fiction films are great for exploring the possibilities. The Island explores the possibility of doing just what I've said here, growing this is a ball of cells and growing it into a whole human being with accelerated growth, so you end up with a clone that's the same age as your patient, which is, as I say, obviously science fiction, but it explored the ethical implications of growing a human being just as a spare part bank, basically. I mean, quite an interesting way of looking at the question.
Yes, and so you would have one cell then to work further with. You can't, artificial conditions continue to...
You grow it until it gets to the blastocyst stage, where there's that inner cell mass inside. Then you disrupt it and you grow the cells as individual cells. You are not allowed, in English law, you're not allowed to keep a human embryo growing in the laboratory for more than 14 days. That's an absolute secret.
My question is not whether it's legal, but is it possible?
Technically, since it's not legal, I don't know whether anybody's waited to see how long they can grow it without it needing some other... I mean, you'd have to experiment to see. Are you thinking the kind of Brave New World scenario, growing babies in bottles?
Yes, I mean...
Yeah. Technically, I suppose you could grow. You'd have to develop, as I say, the right media to grow it in. But as I say, at the moment, there is a very, very strict ceiling on the 14 days. Yeah.
What is the penalty in law for violating that restriction?
I'd have to look that up. I don't know.
Because if it is going to bring that particular scientist, questionable scientist, I would say,
Yes.
a Nobel Prize.
Yeah.
Maybe he or she may wish it.
Published it, it would be in jail or whatever.
I don't.
But then you can go to collect the Nobel Prize. So
Nobel Prize.
restriction is actually
I don't know what the legal penalty is. I'd have to look that up. I mean, the bill would lay it out, but I don't think I've ever actually looked at the penalties for breaking the restrictions.
I don't think the legal penalty is not the first thing the scientists would worry about,
No,
but the end of their career.
Absolutely.
That's what they would worry about. Let's get it clear. Yeah.
But yeah, so it is as I say.
Just make a comment on it. It is interesting that the Human Fertilisation Embryology Act, which came out of Mary Warnock's committee, does have very stringent. It's part of the criminal law, and as I remember, it's 3 years in jail or something like that. So it's a severe criminal offence, and their strategy was to have a very severe criminal jurisdiction within a relatively liberal framework, so that within that framework there was a lot of freedom, but their strategy was to have very severe outer portion and then to use the full force of the criminal law to uphold that.
So a very large field to play in, but an electric fence around the
Yeah,
outside.
that's right.
Yeah, so
Can I say as well that all scientific journals require that things published in them are done according to ethical practice.
Of course,
so
yes.
the scientists wouldn't be able to publish the results, so there's no point in doing the experiment.
yes, thank you. I mean, I think it's good to ask the question though. So thank you for asking. But yes, I just wanted to check, everybody was reasonably okay with what we're doing and what we're talking about here. Okay, let's move on then. So the controversy around this issue, which is the main centre of the talk really, is that the cells, because the cells are taken from this inner cell mass here, which grows into the embryo, effectively you are destroying a human embryo at this stage in order to obtain the embryonic stem cells. And for some people, whether they're from particular faith communities or not, that is completely unethical, because it is using a human being as a means to an end, not as an end in themselves. It's employing them as a sort of an instrument of somebody else's will, rather than seeing them as a person in their own rights. Obviously this ties in with the subject from the last talk. So there are different approaches to this in public discourse, and obviously when I say public discourse, that's including various faith communities' contributions, but we're not kind of looking just at the faith basis today, but kind of the public arena. And one, obviously picking up from this morning's talk, is the utilitarian approach. Do the ends justify the means? Let's maximise the happiness. And this is an argument which is put forward by a lot of people who work in this field. There is much made of the potential medical benefits of research and findings accruing from research on embryonic stem cells. And in case those of you wonder, I'm going to go back to adult stem cells later. But specifically on embryonic stem cells, people would say, well, think of all the benefits. There are going to be lots and lots of people may benefit from the destruction of any one particular embryo. And therefore, especially as this entity, although it's human and nobody is disputing the fact that it's a human being, it's a ball of cells. There is no nervous sensitivity there. We can't be accused of causing this entity any pain, and therefore, on the grand scale of things, it's better to break up this ball of cells and yield potentially wonderful medical advances by destroying this embryo. Within the public discourse, this utilitarian argument comes up again and again, and it's particularly employed by those well-known people who have lent their name to the campaign to enable embryonic stem cell research, particularly in America, where, of course, until recently there was a moratorium on federal funding for it, although it was still legal. But particularly people like Nancy Reagan was campaigning for stem cell research because Ronald Reagan was suffering from Alzheimer's, and there was an idea that stem cell research could eventually result in some treatment for Alzheimer's. You could replace the cells which weren't working properly. Michael J. Fox is a fairly well-known actor in the States who's got Parkinson's, young onset Parkinson's. And Christopher Reeve was very well known for campaigning after his accident where he fell off his horse and became paraplegic, campaigning for the use of embryonic stem cell research in order to find a cure for paralysis and the growing of nerve cells to kind of mend the rupture that had happened in people's nervous systems as a result of accidents and other causes. So there's been a high-profile campaign with people who are in the public eye arguing that this research has the potential to bless so many people that it should be allowed to go ahead and it should be given federal funding in the United States. In contrast to a utilitarian approach, we have the deontological approach, which asks, well, if it's wrong, and this is not necessarily, you might decide deontologically it's okay to kill people, but if you decide it is always wrong to kill someone, then it is wrong to kill one human being, even to save the lives of many others. And therefore, it would be wrong to pursue embryonic stem cell research because it results in the destruction of a human embryo. Similar caution would be argued, and has been argued, by people coming from the angle of virtue ethics, who talk about how if we are using human life at its most vulnerable, and there is arguably nothing more vulnerable than human life at the very early stages when it's just a few cells, if we are using that as a means to an end, if we're using that for the benefit of others without it being able to give its own acceptance of its sacrifice, then what does that say about us as people? What does it say about us as a society if we are not looking after the weakest members of that society? So that's not necessarily saying it's always wrong to do this, but it's urging a cautious approach. It's urging society to think about what it does and urging people to think about what they do when they instrumentalise human life in this way. Within faith communities, interestingly, you get some marked differences with regard to stem cell research, much larger differences than you get in the issue of abortion, for instance. And I'm focusing here on the Abrahamic faiths. When it's thinking about abortion, although attitudes vary, there's generally a presumption that abortion is not a good across Jewish, Christian and Islamic scholarship. That some of them would advise that it was allowable under certain circumstances, but it's generally, the general presumption is that it's not a good. However, when it comes to stem cell research, there's quite a different approach. Within the Christian tradition, you will get some people arguing that the very early human embryo is a person in the full sense, and as John was saying earlier, and therefore we should not be destroying this early human embryo. There's an argument, there's a question here on whether totipotency, whether that ability to grow into a full human being means it has exactly the same dignity as the lady next door. Is that kind of, weighing up people at different stages in life? Are we really saying that when it comes down to it, the life of this fertilized egg is as valuable as the person you walk to school with or the person you teach? And there's all sorts of questions there about relative valuing of human beings, but some people would say yes, it is. Other Christians would take the viewpoint that actually human life starts as a fertilized egg, but in fact it doesn't realize its potential until it's implanted in a woman's womb, and therefore from life in the womb should be protected, but those first week or two of development don't have the same kind of protection, and therefore we shouldn't view it in the same way. And as John alluded to earlier, some people will refer to, some Christians will refer to brain death as being the definition now of when you're dead. When your brain's dead, you're dead. In the same way they would look at the beginning of developments of the nervous system as a guide to when we could start talking about a person being there. Before that it's just a ball of tissue. After that, when it starts taking human form, then we should accrue it much greater dignity, and that therefore the kind of beginnings of differentiation of tissue, beginnings of specialization in nervous tissue should be the point at which we start according the human embryo full human dignity as a person. So within the Christian tradition, you'll find proponents of each of those positions. In relation to this, you'll find particularly in thinking back again about the use of the Bible, some people will refer to passages in the Bible where the psalmist or the prophet talks about being known by God in his mother's womb, and will therefore say, well then God obviously cares about life in the womb, and therefore that this is a clear indication that pre-born life is full life in that sense. Others will point out that Jeremiah says, God knew me before I was in my mother's womb, and suggest that therefore in fact this kind of language is more poetic and is not something to be used to argue about embryology. That is, the prophet is talking about God calling him and this sense of God's plan throughout the ages being fulfilled, rather than making a statement about ascribing full human dignity to the very early stages of human life. So debate about biblical passages use in this respect, and debate too about the passage in Exodus where the two men are fighting and one hits the woman, and if the woman miscarries there's a fine, but if she dies it's eye for the eye, tooth for the tooth, life for the life. If she's injured, there's a real kind of penalty to pay. If the child is lost, then there's just a fine. and this maybe this is saying that the life in the womb is not worth the same as the mother's life. This is a kind of relational thing here. Others point out that for Christians particularly, it's not just the Old Testament they're looking at, but it's the New Testament. And if Christ was incarnate, if Christ was conceived in Mary, then it's that stage of life which is dignified by the presence of God, and therefore all stages of human life from fertilization onwards are dignified by the incarnation. So different people will employ different arguments based on different parts of the Bible and different bases for that. In contrast, Jewish commentators they'll talk about the Exodus passage and that it apparently accords a lesser value to the fetus than it does to the mother, that there's a fine for the fetus being lost, but there's a much more severe scale of penalties if the mother is injured or dies. And they also rely a great deal on the extra-biblical interpretation and comments on the biblical text that have built up over the centuries within the Jewish faith. And particularly, one of the references in the Babylonian Talmud is to the developing human embryo, we would now call it, being as if it were just water for the first 40 days. It doesn't have form, and if a woman, I think there's some kind of legal understanding that if a woman was less than 40 days pregnant, she wasn't in the same status regarding her ritual cleanliness of her pregnancy as she was once she got past 40 days. That was a significant point in the pregnancy at which the way she related to the rest of the community changed. They also point out that under Jewish law, a child does not have the same rights as the mother, equal rights to the mother, during a pregnancy until either the head is showing or at least half the body if it's a breech birth. Up until that point, although they would frown upon abortion, if there's a straight choice between the mother and the child and the fetus, it's always the mother's life takes precedence over the fetus. And even once the child is born, it is not taken fully into the community for a few days. And if the child seems to be early, they'll leave a period of 30 days before they'll really think the child is safely born, if you like. It's not going to die prematurely. If a child dies before, I think it's 30 days, the Jewish community is not obliged to sit Shiva for it in the same way as they are if the child dies at a later stage. So it's almost like a sliding scale of responsibilities of the community towards the child. And because of these teachings and these attitudes, particularly this one, the Jewish community sees stem cell research as a means to save life and would encourage, certainly the Jewish commentators I've come across, would encourage embryonic stem cell research. Again, there's a variation, the same as there is within the Christian tradition. And again, there's another variation across Islamic commentators. There is similarly this teaching, there is a certain amount, not a lot, but there's a little in the Quran and some in the Hadith, the sayings of the Prophet, which seem to indicate that there are stages of 40 days in the development of the human being in the womb and God's interactions with it. And certainly most jurists, most Islamic jurists, give a particular significance to the fact, again, you've got this reference to a water drop. The person is as a water drop for 40 days. Which again has been used to suggest that from an Islamic point of view there shouldn't be any problems with embryonic stem cell research. Much, and later again, they're not encouraging abortion or anything like that, but certainly from the point of view of stem cell research, at this early stage they don't seem to have any problems. And again, you will find people whose opinions vary. I'm not saying this is the Islamic position or the Jewish position any more than I could stand here and say this is the Christian position. But it just illustrates how different religious communities have different texts and they're employing in different ways to come up with a varying number of approaches towards this question. I think it's interesting that the religious opinions are in some cases affected by the developmental biology. That I wonder how many people know very much or knew very much about the early stages in human development. And I think that now people have seen pictures of developing embryos, and you can see the child in the womb with some of these amazing new methods of visualizing intrauterine life, that I think somehow the scientific findings have affected public opinion and how they think about life before birth. It is, as James Gustafson says in his book on science, theology and ethics, explaining and valuing the human is where you get an inevitable theological and scientific encounter. You can't think about this topic without knowing some of the science and using that to reflect on theologically. The two go hand in hand. So we've got this, if we have the personhood of a human being from fertilization onwards, and by the way, I'm not using the word conception deliberately, because I think technically conception is really the whole process of fertilization up to implantation, which is when the woman is actually pregnant, because it triggers off the body's reactions to the developing life. So I'm deliberately using fertilization here rather than conception. Some people, though, argue, well, wait a minute, we can use adult stem cells to achieve the same aim. And there, you'll be aware there's been a lot of discussion recently about adult stem cells being used, not just because they can be persuaded to give a range of different cells, but we can actually, as it were, now persuade adult stem cells to open up some of those recipes they'd shut. We can pull them further back down the path of specialization and make them less specialized. And there's been a lot of work over the last 2 years. These papers came out last year, and the term is induced pluripotent stem cells. So they're pluripotent like those cells from the inner cell mass, but they're induced from more specialized cells. In this case, adult fibroblasts from skin, and actually they both used fibroblasts. These particular experiments last year suffered from the drawback that they used viral vectors to put genes in to make the cells, to kind of make some more of the structures of DNA available, if you like. And as we heard earlier today, talking about the bubble babies who had had gene treatment, and then they discovered the genes that inserted into other cells, into other genes, which made them susceptible to cancer, and some of the children had gone on to develop cancer. Putting viral vectors in is always risky, because it's like trying to shoot a missing word into a copy of Shakespeare's works and hoping that it doesn't end up being printed in the middle of another word. It's that kind of risk. You might mess up another part of the story by putting in the word at random. Very recently we've had some more, actually, that's a bit further on, I've got a little bit more on some more recent work on that. In terms of the public discourse, so we've got the debate about the personhood of the early embryo, we've got the possibility of using adult stem cells, we've also got this kind of the yuck factor again. We saw some of those things this morning about frankenfoods and stuff like that. You get the same kind of ideas in public discourse on this, and even with looking about stem cell treatments that were in, this was in the press last week, British scientists create human sperm from stem cells, and the comment on the next page of the Daily Telegraph, man at his maddest. Obviously the Daily Telegraph writer hasn't listened to us about using gender inclusive language, but I guess they thought with sperm it was good man at his maddest. So you do get, public discourse does have this, again, this kind of, is that right? Yes, Leon Cass, who was the head of President Bush's Council on Bioethics, referred to the yuck factor as the wisdom of repugnance, which I thought was an interesting way to put it, the wisdom of, if you get this kind of, oh, really, gross, is there actually, there's a wisdom in there that you would do well to listen to and not kind of suppress that reaction. Thinking very briefly about the legal policy regarding embryos, we touched on this a little bit earlier in response to questions. With the things that we can now do in the laboratory that are now legal in Britain, we can use egg cells from, not from the mother, but from other women in order to avoid mitochondrial disease. Some women have a condition whereby, if you think about the human egg as the kitchen again, they've got all the recipes, half the recipes there, but some of the saucepans have got holes in. Some of the stuff in the egg cell, the mitochondria that should be working to produce the power that the cell needs, have a problem in their DNA. There's a very little bit of DNA in mitochondria, and there's a problem with that in some women. But there's nothing wrong with their genetic material. Most of the DNA is stored in the nucleus. Sorry, the nuclear material. There's nothing wrong with their nuclear DNA. So if you can get their egg fertilized with their partner's sperm, but then take the recipes, if you like, take the recipe books out and put them in another kitchen, an empty kitchen, an enucleated egg, like we saw in one of those earlier experiments, that we could help treat, so women with mitochondrial disease could then have healthy children with another woman's egg cells, and I'll talk about that more later if necessary. Oops, sorry. You can create human embryos for research purposes, including stem cell development now, whereas before it used to be that they could only use spare embryos from IVF, ones which people had donated after, if they finished their IVF treatment and they got enough children and they didn't want the spare embryos to be left in the freezer, they could donate them to science, but now they can create them specifically for research purposes. And there was this provision also in the bill last year to allow human admixed embryos, which was a kind of rather catch-all phrase for putting some genetic material from humans into animal embryos, or making chimeras of human embryonic cells and animal cells in various mixtures. But again, 14-day limit, absolute strict limit on how long you could keep them from their initial creation, 14 days of development maximum, and then you have to destroy them. The bill last year also allowed a greater range of conditions for which you could select embryos by pre-implantation genetic diagnosis. I don't know whether, when people hear about pre-implantation genetic diagnosis, they realise that it is really a very gentle eugenics, because instead of sending them all off to gas chambers, it's a case of finding out when the egg is first fertilised, when you've got that ball of cells, does it carry an undesirable trait? Well, we'll throw that one away then. So effectively, the pre-implantation genetic diagnosis is having a profound effect on the sorts of human beings that are being allowed to be born by IVF procedures. And you'll find some disability groups are campaigning strongly for an ending to some diagnosis techniques, because they say it's discriminating against the disabled, because if you're virtually saying they shouldn't have been born, if you're eliminating them at this very early stage. That's another kind of slightly side issue to stem cells, but was covered in the bill last year. And one of the other main parts that came up in the bill last year was the kind of the definition of parent, whether you could have 2 women or 2 men as the named parents on children conceived using IVF. So, just drawing to a close now, the development of embryonic stem cells has been controversial since it started. I think there's, within Britain, because the legal situation has been much more flexible than it is, not just, it has been not only in the United States but also in other parts of Europe. There's a lot of this research has gone on in England. There is a general public acceptance of it, although there was a big debate last year, and that some of the church communities played a large part in the public debate about the bill. The bill was nevertheless passed. There is pressure now from some faith communities to encourage their members to campaign politically, again, for their MPs to try and stop there being any more change in the law and to try and get some of the existing laws changed. This is an example taken from the Roman Catholic tradition, but you would get maybe less formal documents. You would certainly get in some other Christian traditions the same kind of exhortation to people to work for the reform of what they see as undesirable legal frameworks for this kind of work. So in terms of public policy and faith communities, there's very much still an ongoing debate and a desire on the part of many faith communities to have an influence on the legal frameworks for this kind of research. There's also a continuing effort to change hearts and minds in the wider public discourse. Interestingly, I find, I know John had this when, I can't remember the exact phrase, where is he? The exact phrase you used about, was it when...
At what point can I say that this is not me?
At what point can I say that this is not me? And he had a series of photographs of an increasingly young human being on the screen. You will find this kind of picture gets employed a lot in campaigns to argue against embryonic stem cell research. This picture doesn't seem to get used as much, even though this is kind of closer to the stage we're talking about. And I do wonder how much, as human beings, maybe evolutionarily, we are conditioned to respond to something that looks like us. That there's this kind of about something that has something that's recognisable, head and arms and eyes. And it's much harder, I think, for us to empathise, to feel identity with this. Maybe that's just me. Somebody in one of the newspapers, again, when this research was going through, one of the comments in one of the British newspapers was exactly what I said earlier. He said, I understand that people are concerned about human life, but I cannot think about a ball of cells in the same way as I think about my next door neighbour. He says, with the best will in the world, I cannot equate the two in my mind. If I had to choose between one or the other, I would always save my next door neighbour in preference to a ball of cells. So I think there is something very fundamental about the way that we engage with this topic. What I meant to say earlier, and I didn't get round to, but I always like to make clear when I'm teaching about anything in this area on IVF or stem cells, is that my husband and I have been in the situation where we have suffered from infertility and we haven't been able to have kids. So just in case, I know sometimes one might think, oh, well, she's up there talking about this and she hasn't got anything vested, anything personal in this, but I just feel it's fair to let you know that this is something that has been quite personal to me, because when you've been through the kind of infertility roundabout medically, this is not just theory, this is something that actually you do think through in some detail. Just thought that was fair to throw that in so that you know where I'm coming from. And just finally, to muddy the water still further, we have this very recent paper with a much safer way of inducing pluripotency in adult stem cells. So this is using virus-free method, and we see here again, they got human cell lines with robust expression of pluripotency markers. They're using embryonic fibroblasts with robust expression, but what they're trying to get is a system from which they can leave cells that are going to be usable for medicine, drug screening and the establishment of disease. It's much safer if you're not using the viral vectors. So this is a big advance in the possible development of adult stem cells to provide alternatives to using embryonic stem cells. However, just to finish, a point which Ted Peters made some years back, and which I always find very thought-provoking, if you can take one of your skin cells and you can treat it with a more sophisticated yet version of the treatment here, and you can make that cell unspecialise back to the stage where it's pluripotent, What about if you could make that go back a bit further until it was totipotent? Would that mean that your skin cell was a potential human being? And if so, how would you think about your skin? Thank you very much.
Thanks. I think we'll have perhaps 10 or 15 minutes of questions, then a slight pause, then we'll have the panel. Do write questions down for the panel during this time, put them in the box. Thank you. So it's open for questions. Yeah.
Thanks a million. Fantastic. Just a quick comment first. I thought the fuss about the sperm, I may have completely misunderstood, but I thought at least part of it wasn't so much the yuck factor, but like male journalists at the Daily Mail were worried about becoming redundant, you know. I thought that's
In
what
more
I was
senses than one, yes. Yes.
One question on IVF. Is it not a utilitarian, I may have misunderstood, is it not a utilitarian argument that in IVF, as I understand it, the embryonic cells that aren't used are quietly eventually disposed of?
Yes.
So why not use them anyway? Is that not an argument?
Yes, I mean, and that has been an argument which I suppose because the recent bill had allowed people to make them deliberately for research purposes, to make embryos deliberately for research purposes, I had kind of skated over that. But yes, you're right. In terms of spares, people, they're not allowed to keep spare embryos on ice without specific permission from the parents. Obviously, technically, they belong to the parents of the embryos at that stage. If the parents have decided they've completed their family and they don't want to use anymore, then they have to be either destroyed or they can be donated to science. And so, yes, from that point of view, you're right, there is a utilitarian argument to say, well, if they're going to be destroyed anyway, then let's use them for research purposes. Yes, it's a very good point. I should have made that one. Thank you, yeah.
I'll just add this, that in our Christian community in Los Angeles, that we would never donate any of the eggs. All eggs would absolutely be implanted. My wife and I have actually gone through IVF as well, and it's just our conviction as exhorted by our pastors, and we agree that under no circumstances would we allow eggs to be donated for science. I can also mention that I can relate to you in the discussion of IVF. When my wife and I went through that, there were two eggs took actually, and as it turns out, one had Patau syndrome, the other had Down syndrome. And those are the only two that actually survived that initial stage. And I can just say that it was sort of crushing to us on one hand. We had to really pray about that because my first reaction was, just in my heart, is I want to start all over again. And I can tell you that when I searched my heart and thought about Christ, I realized that, as was said earlier today, I've never personally met a handicapped child who's ever said, I wish I were dead. And I realized, frankly, personally, the breadth of my selfishness. So my wife and I had those implanted because we realized we could not, with Christian integrity, not do that. As it turns out, both perish. But just to make you aware that there is a very strong, and there are organizations in California, adoption agencies, that specifically focus on the adoption of those unused eggs.
Yes, is it Snowflake?
Yes.
One of them? Yes,
right.
yeah. Yes, I mean, if I'm talking about IVF specifically, I mean, I didn't today because we were trying to stick on the stem cells, but yes, you're right, certainly with IVF, there are agencies that will adopt spare embryos if the parents are agreeable. And that is one way to avoid the kind of the throwaway disposable. And also, I mean, certainly some doctors in this country will offer the kind of, well, we'll only fertilise the number that we could put in so that there won't be any spares. So, yeah, I think some medical personnel are very much open to the ethical dilemmas that are thrown up by IVF. Some, in my experience, are totally oblivious to any ethical. And one doctor very calmly said, oh, well, there's 7 eggs there this month, you know, but if they all get fertilised, we'll just selectively destroy some. Don't worry. Without any kind of by your leave or how do you feel about this or anything. So I have experienced the very good side of the medical profession and the extremely cack-handed side of it as well. So, thank you.
can you just, in the light of that, can you just reflect on the fact that in nature, if you like, in the norm system, huge numbers are discarded?
Yeah,
are
absolutely,
lost.
yes. Yeah.
So, are we coming out at the right angle when we worry so much when the system normally doesn't?
I think, yeah, that's a very good point, and that's an argument used a lot in reflecting on the status of the early human embryo, that, and as what I said earlier too, that from my point of view, I feel I think John said earlier, the woman is the mother from that point when fertilisation occurs. I would disagree with all respect and say that she's not a mother or potential mother until implantation occurs, and that's, in my understanding, that's when the pregnancy starts, and that early stage is still that kind of hit and miss. It's in the same category as will the sperm find the egg kind of, or is it the right time of month kind of category, where there's so much chance involved. And as you say, a lot of fertilised, we believe, is it something like 40% or more of fertilised? Don't think people can be, yeah. It's it's difficult to be sure, but certainly a lot are lost at that early stage.
I can see lots of hands going up, so we must move across.
Just to comment on that, I mean, it seems to me a fallacious argument, because millions of children we know will be dying in the third world from preventable diseases, but we don't then say children aren't important, do we?
Right.
No, I wasn't making any argument, I
No.
was just asking for a reflection on this fact.
And I think too, there's a very real, as John pointed out earlier, it is in a sense ridiculous that up to a certain time, because the child is in the womb, we can legitimately kill it under certain restricted circumstances, but we can still kill it. Whereas the day after, if it happened to be born, then it would be totally against the law to kill it. And how that difference of whether it's in the womb or not affects its legal status in such a remarkable fashion seems to be an anomaly to me in the way that the law's set up.
So we'll move on across it.
I possibly comment on your last statement, that it seems to me that it is the nature of the law that obviously it's a responsive system. It's not something which is designed overnight. So therefore, the definition in law of a person, if you actually go back in history, is someone who actually was born. So if before you were born, you're not a person, quite correctly in most legal systems I'm aware of. So that I think partly addresses the problem. Why? A day ago, a child which was not born yet is not a person, so can be killed. And then a day later, well, nothing has really changed in a day, except that
It just seems a little. I agree with you totally. The law is law. The way it's formulated, it has to have hard and fast cut-off points. But it can still seem a little bizarre. And obviously, as I said again earlier, with this thing with if the mother's life is endangered by the child in Jewish law, as I understand it, and I'm not an expert, I'm totally dependent on Jewish sources for this, but as I understand it, the mother's life would take precedent, but if the child's head is out, or half the rest of it, then it doesn't. The two have equal, it's at that point, there is this very clear understanding that something profoundly new is present there, and there is that, I guess it's because it's expressive of the independence of the child. It's not dependent on the mother's body for life anymore.
Your illustration of the skin, if we can use skin, we're shedding skin all the time. Does this not indicate to us that we're being a bit neurotic about this early sensitivity to, I think it's 80% of it of fertilized eggs is naturally rejected by the body because there's something not quite right about them. So I don't see the agonizings over possible rejection of a fetus that we've detected there's something seriously wrong with it.
Yeah, do you mean the last example where I said about you can take a skin cell and make it to, that's a bit of a thought experiment that Ted Peters was questioning whether we really want to say if something is totipotent, it's a person. Are we saying that it equals human dignity, that it's the potency of that particular cell that determines its human dignity, whereas a stem cell, because you can't get a baby out of it, doesn't. Are we really saying that that's all there is? That's the difference between them? And it's deliberately made. I think the thought experiment is deliberately made to sound a little ridiculous, to provoke that kind of thought about that. And I think it's worth serious consideration, even though, as you say, it can sound a little ridiculous, but I think it's highlighting a very important question.
Yeah, thanks a lot for your talk. It was very, very enlightening. My question is really, it's related to the previous couple of questions, is that it strikes me that whether this is a human person or not, I mean, maybe that is debatable, but at the very least it's a human life. We have the life of a human being. It could potentially become a person, and so a human person can potentially. And my question is, should we not perhaps invoke the precautionary principle?
Yeah.
It's different from nature discarding eggs, because I think that's a natural process. But what we're doing here is human beings and scientists, often for very specific reasons to do with funding, profits, commercial exploitation, etc., are interfering with the very mechanisms of human life itself. And whereas that we often don't really know what the consequence might be in a very delicate area of our existence. And I think, and many of the claims, like for cures, etc., coming out of embryonic etc., apparently there hasn't been one cure yet. Would that be the case?
As far as I know, the only treatments that are actually being tried at the moment are derived from adult stem cell treatments. So I might be corrected by
I think that's probably
my colleagues.
But so my question is, the ethical issue here, and I think it is an ethical issue as well, is should we not invoke the precautionary principle here in allowing this kind of research? And it strikes me that the bill that was passed by the British government was completely unethical, actually, because it did not really take into account that precautionary principle.
Well, I suppose you could say they've been precautionary in that they were allowing it for experimental purposes, but they weren't, and there's very much this 14-day limit, and they were, but I think the difficulty is that with even people who admit that adult stem cells are proving perhaps more successful than they'd anticipated in generating potential treatments, medical treatments, some of them would still argue that it's worth doing some experimentation on embryonic stem cells because they are means of helping us work out the factors we need to play with in order to get cells to get from this unspecialised state down to a specialised, whatever end state we want, whether we want kidney or skin or something else. And so they would argue that we still need to experiment on embryonic stem cells. There is also related to what you said about the precautionary principle, the problem that, the thing about stem cells is, A, they have the potential to give more specialised cells, and B, they keep dividing. That's why they're stem cells. They have the potential to keep dividing and replenishing themselves as well as providing other cells to specialise. And therefore there is always going to be the potential danger of development of tumours So a lot of what they're working with is trying to check when that's going to happen and what's safe to put where. And one of the reasons they want to mix human embryonic cells in with animal embryos is because they don't want to do experiments with human beings before they've checked out the safety of some of the techniques in animals. And if they put human embryonic cells into animal embryos, they can see how they behave in an embryo, and they can get some idea about what sorts of behaviours to expect from stem cells if you put them in an unusual circumstance. So a lot of that is precautionary in that they want to do the experiments then and find out what the dangers are. So that would be an argument that would be put forward, I suspect, by somebody working in embryonic stem cell research. I mean, it's not my research field, so I don't have that vested kind of, this should carry on kind of feeling about it. But certainly, I think some of the people who are doing the research, it isn't just, of course it's fun, let's see what happens if we put this and this together. There is a real desire to benefit people and to provide the safety measures and the kind of conditions right. So their precautionary principle, in one sense they might say that's what they're trying to follow. They're trying to make sure it's safe before they use it. But it's a good point.
I think I'm going to just call a halt to the
because
questions
we've got
now.
time.
So that